Hello, my name is Ariel Wolcher from the Nazeer lab at SUNY POTSDAM, and today I'm going to be talking with you about the effects of transcription blocking drug on mRNA processing factors. Now, the goal of our research was to test what was occurring after we induced DNA damage via UV radiation. We wanted to test to see whether it was DNA damage that was occurring or whether it was RNA polymerase II stalling. Now first, I'm going to explain what transcription is really quick. So transcription is a process that creates messenger RNA from a DNA template. And basically after the mRNA has processed, we create mature mRNA, which translates into proteins that are used by the body for various functions. So we are looking at how transcription is moving along at that DNA to mRNA process. And to do that, we are actually looking at different processing factors and how they are affected by either having a reduced number of these processing factors or whether they stay pretty much the same. To do this, we used a treatment called 6-AU or 6-azauracil, which actually stalls RNAP II, RNA polymerase II, which can be seen in our background right there. And we went and we took these and we analyzed the mRNA processing levels with a Western blotting procedure. We didn't really see any reduced levels of mRNA processing factors with this treatment, which suggests that we might be observing an inhibition of mRNA processing, and lower levels of processing factors are a result of DNA damage instead of RNAP II stalling. So our hypothesis is that certain proteins are responsible for the degradation of RPB1. These include DEF1, ELK1, and RPB9, which are involved in blocking mRNA processing. So we used different strains. We used W303, treated and untreated, a strain of RPB9, treated, untreated, DEF1, treated, untreated, ELK1, treated and untreated. And we tested the proteins RPB1, YSH1, RAD53, actin, and SUZU72. So actin was primarily our control. RPB1 is actually a subunit of RNAP II, and it is the largest subunit, and it is degraded following DNA damage. And RAD53 is phosphorylated in response to DNA damage. And YSH1 and SSU72 are mRNA processing factors that are required to form the 3' end of transcribed RNA. And we did not see a change in the levels of any of these after treatment. So none of the cell lines showed RPB1 degradation or shift in RAD53 on the gel. And this suggests that 6-AU did not induce a DNA response. And then for mRNA factor levels, we saw them completely unchanged in all cell lines tested, whether they were treated with 6-AU or not. And this suggests that the RNAP II stalling without the DNA damage did not result in a degradation of the mRNA processing factors. So to do this, we used bacterial transformation, which was cells were transformed with a plasmid that allowed the cells to grow on a media lacking uracil. And we used various heating and cooling stages to do this. Various types of water baths were used in that procedure. We also grew the cells on negative ura media, and we treated them with 6-AU. We also did cell extract preparations, which were prepared using lithium acetate, NaOH extraction procedure. and then they were left to grow overnight and centrifuged and treated with lithium acetate and NaOH again. And then the cell debris were removed, and then they were extracted using Lameli buffer. And then we also did a Western blot analysis, which allowed us to run gels, which were loaded with the subjects being tested. And then we transferred them to a membrane. And then the membranes were treated with the primary and secondary antibodies. And then we also did a chemoluminescence, which allowed us to see these after they had been transferred. And so our further directions are to analyze for other processing factor levels in these extracts and complete our analysis of the mRNA processing factor levels following 6-AU treatment. Thank you for your interest in our research, and we'd also like to thank the Kilmer Foundation. Thank you.